Welcome to Unit 2 of the ESPT training. This unit builds on Unit 1 and covers sampling methods, site and survey selection, sampling design, and entomological data management. Now we will start with Module 3 on selecting sampling methods and analytical techniques. As a reminder, this training module corresponds to Module 3 of the ESPT. By the end of this module, you should be able to compare the advantages, limitations, and biases of each entomological sampling method and analysis technique. Use the ESPT to select appropriate methods to address your priority program question or questions while accounting for method limitations and biases. And finally, use the ESPT to select the appropriate method for assessing human behavior and high-risk populations as this relates to human vector exposure. Before we get started, let's revisit our training checklist. First, we recommend that you have either a digital or paper copy of the ESPT in front of you. And please also have a computer or notepad and writing utensils such as a pencil or a pen. We ask that you retain your program question or questions identified in Module 1 as well as your indicator selected in Module 2. Remember that each module builds on the previous module. VoiceOver is recommended. The orange background slides indicate our illustrative example, and finally, the gray background slides indicate an applied exercise that can be done individually or as a group. You should plan about 10 to 20 minutes for each exercise. At this point in the ESPT training course, you should have defined your priority question by working through Module 1, and you should have selected the appropriate indicators to address your priority question. If this is your first time working through the ESPT, it's best to work through Module 3 only after you have worked through and understood Modules 1 and 2. Now, we will explore how Module 3 helps you determine the appropriate sampling methods to collect data on your selected indicators. This module summarizes common sampling and lab techniques in easy-to-use tables. Let's begin. In the following 6 slides, we will first discuss how Module 3 helps you select the appropriate sampling methods and laboratory techniques to collect the data that will address your priority question. Field sampling methods must be selected based on your minimum essential indicators. There are 2 key points to bear in mind during the selection process. The first point is that entomological sampling methods take advantage of specific mosquito behaviors. For instance, human landing catches target and capture mosquitoes that are host-seeking. Similarly, pyrethrum spray catches capture mosquitoes that are resting. You can refer to Table 8 of the ESPT, which lists specific trap methods and the mosquito behaviors they each target. The second point is that each method comes with biases, advantages, and limitations. For example, the HLC might be most effective for measuring human biting rates, but it is also expensive and labor-intensive. You can refer to Box 1 for a list common sampling techniques, many of which you might have experience using yourself, such as the CDC light trap and HLC. If you'd like more information on these sampling methods, please pause the video now and review Annex 3 of the ESPT. When selecting multiple sampling methods, consider how they may interact. For instance, residual pyrethrum from a morning PSC may inhibit mosquitoes from approaching an HLC collector the following night if these 2 methods are used in the same house. Likewise, an HLC collector prevents mosquitoes from resting by removing them from the population. Therefore, a PSC performed at the same house the night after an HLC is likely to underestimate indoor resting density. Because of these interactions, different houses should be sampled for HLCs and PSCs. In addition, always try to optimize your sampling plan. Well-designed sampling can capture data from more than one indicator. For example, indoor and outdoor HLCs can generate data to determine species composition, biting behavior, and biting location. Table 8 in Module 3 helps you select appropriate methods based on your minimum essential indicators. On the left, the second column lists common entomological sampling methods such as HLCs, CDC light traps, and larval surveys. The third column lists the mosquito behavior each sampling method targets, while the fourth column indicates whether the method is biased towards mosquitoes with a particular host preference. Directly to the right of the fourth column are a series of minimum essential and supplemental indicators. When a given method collects data relevant to measure a given indicator, the corresponding cell is colored in teal. Finally, the last column to the right provides examples of common trap types used for a given method. Use Table 8 to help you determine what types of questions and entomological indicators each sampling method may help you address. For example, let's say I'm looking to measure biting time. Based on the table, I see that I can use several methods, including the HLC method by conducting HLCs inside and outside. I can also see that there are several methods I can't use, such as indoor resting collections and outdoor resting collections. Table 9 in Module 3 summarizes the limitations, advantages, and biases of common collection methods. A good time to use this table is when you are planning how to conduct your entomological fieldwork, and you must make decisions about which methods to use given your available capacity and resources. The first column to the left includes the same sampling methods listed in Table 8. Directly to the right of this column are a list of trap type examples. The following series of columns to the right include characteristics of each sampling method that may help determine whether it would be appropriate for your context. For instance, each method is rated for its level of difficult, 1 being the easiest and 5 being the most difficult. The second to last column indicates whether a given method can be used to help you determine whether or not IRS, LLINs, or larviciding might be appropriate interventions. For example, you can see that HLCs are suitable to assess the appropriateness of LLINs, but not of IRS and larviciding. Finally, the last column indicates whether a sampling method is appropriate to evaluate current interventions in use. Next, we will move on to selecting lab techniques, but we will come back to these tables in a few minutes so that you can see how we use them to select our sampling methods for our example question. Once vectors have been collected, they are usually brought to the laboratory for analysis. Let's discuss how to select techniques to analyze mosquitoes in the lab. The data required to calculate many indicators are generated by standardized entomological lab techniques. Box 2 highlights some of these techniques, such as PCR for species identification and assays for insecticide susceptibility testing. Each of these techniques has biases, similar to those related to sampling methods we discussed earlier. For example, the morphologically indistinguishable Anopheles gambi complex has multiple species with diverse behaviors that contribute to disease transmission differently. Restricting your laboratory techniques to morphological identification will affect the accuracy of vector species data. Many of these lab techniques, such as PCR, require appropriate training and infrastructure. When national malaria program capacity for this type of work is limited, collaboration with local or international partners may support these activities. To appropriately target vector control interventions, it is critical to know which humans to target and when and where they are exposed to mosquito bites. In Module 2, we learned how data from HBOs can be integrated with data on vector bionomics to calculate the adjusted biting rate indicator and therefore identify gaps in protection. There are additional human-centric data collection methods for identifying high-risk populations. These include formative assessment and case control studies. These methods are beyond the scope of this training, but you can find further information on characterizing HRPs in Box 3 of the ESPT and in our dedicated HRP tool on the MEI website. Great, we have now finished going over Module 3's content. Now let's look at how Module 3 helps us select the right methods to collect data on our chosen indicators, which are vector occurrence, biting location, biting time, human biting rate, and insecticide resistance frequency, and therefore answer our example question: are bed nets an appropriate intervention for the village community of Katosha? Here you will see how Table 8 can help us select the right sampling method for our example question. Are bed nets an appropriate intervention for the village community of Katosha? Recall that the 5 indicators we decided to measure to answer this question are vector occurrence, biting location, biting time, human biting rate, and insecticide resistance frequency. We actually have 2 more indicators: adjusted biting rates and human sleeping behavior, but we'll return to discuss those in more detail in the next slide. Looking at this table, we can immediately see that there are several sampling methods that allow us to measure these indicators. In fact, HLCs, human baited traps, and CDC light traps each allow us to measure all 5 of our indicators. Indoor resting collections would allow us to measure only 2 out of our 5 indicators. So this is probably not the best option. Now we need to decide between HLCs, HBTs, and CDC light traps. HLCs are the gold standard for measuring human biting rates, but they are labor intensive and can become expensive. CDC light traps are less laborious and less costly, but after an initial trap validation in Katosha comparing the HLCs versus CDC light traps, we saw that CDC light traps did not collect nearly the same amount of Anopheles as HLC. So we concluded that CDC light traps were not a valid proxy for HLCs in Katosha. So our remaining 2 options are HLCs and HBTs. Our ethics board approves of HLC, so we will select HLCs. In the previous slide, we decided to select HLCs as our sampling method of choice. We decided this because HLCs allow us to measure all 5 of our priority indicators. Our last 2 indicators were adjusted biting rates and human sleeping patterns. Since HBOs are easy, inexpensive, and the only standardized way to calculate adjusted biting rates, we decided to use this method to collect data for these indicators. Here is a summary of what the data shows. Between 10 and 11 p.m., the collectors find mosquitoes both indoors and outdoors. The collectors also count people indoors, sleeping under their bed nets. They also count people outdoors, socializing. Between 10 and 11 p.m., there is no exposure to vector bites because people are protected by their bed nets. However, there is exposure to mosquito bites outdoors because the people socializing outside are not protected by any intervention. Based on this data, we can already determine that bed nets have a role in Katosha. If there had been no bed nets, people would have been bitten by the indoor mosquitoes while sleeping. Now let's move on to our first exercise in Module 3. You can work on this individually or as a small group. For this first exercise, you will need to have your priority question from the exercise in Module 1 and the minimum essential indicators from the exercise in Module 2. We ask you to identify which methods are most appropriate for your indicators. To answer this question, use Table 8 to select all the methods appropriate for your indicators. Then narrow down your list using Table 9. Select 1 or 2 sampling methods you think are most appropriate for your question and context. Keep in mind how available capacity might affect your decision. For example, please pause the video now. Once you are done, we can move on to the second participant exercise. Now let's see how you could integrate human behavior and or HRP survey methods to address your question. But first, you'll need to determine if human behavior is relevant. Then if you decide it is, use box 3 to help you decide which human behavior survey method is most suitable for your question and context. In 3 points, briefly outline how you would conduct both vector and human behavior or HRP surveys. Pause the video now. Once you have completed this exercise, let's proceed to the final slide of this module. This completes Module 3 and Unit 1 of the ESPT training. At this stage, you should be able to identify and formulate a programmatically relevant question, select appropriate indicators to answer the program question, and select methods that provide the data required to calculate your indicators. Up next is Module 4. This module will cover site selection and survey types, and we invite you to proceed to that module now. On behalf of UCSF's Malaria Elimination Initiative, thank you for listening, and we hope that this was a productive session.